Topic of presentation is basic muscle structure. Okay, there are around more than 400 muscles are present in our body. More than 400 muscle, and muscles are mainly three types: skeletal muscle, cardiac muscle, as well as your smooth muscle. Among all three, skeletal and cardiac muscles are straighted muscle. Straight muscle means they are having a straight fibers. Fibers are going straight, whereas smooth muscle doesn't have this type of property. And only your skeletal muscle forms around 40 to 60% of your body weight. Clear? Now we will discuss the gross anatomy of muscle structure. Gross structure, anatomy of muscle. Muscle are a rope-like structure. You can see a rope. You can imagine ropes. If you closely observe a rope, ropes are made up of a very thin fiber. Many thin fibers wrap around, make one more thick fiber, then one more. By rearranging many thin fibers, you can get a resulted thick rope. Same as with the muscles are, when you see muscle, when you cut muscle grossly, you will find three connective tissue sheath. They are not muscle fiber, they are basically covering the outer covering sheath which are surrounding the muscle fiber. Like you can imagine a electrical wire, a big wire, black cable. The plastic is act as a sheath. The inside is your copper wire. So copper wire, you can imagine copper wire as a muscle, but the plastic sheet, red, green, whatever, or black or yellow, whatever, is acting as a sheath. So the muscle fibers, very thin fibers, are covered by, many thin fibers are covered by a thick insulation is known as endomysium. I will show you in diagram. Endomysium is the innermost covering of innermost connective tissue sheath which cover the muscle fiber, main fiber, the thinnest portion of muscle known as muscle fiber. And muscle fiber, many muscle fiber, all muscle fibers are encapsulated or wrapped by a connective sheath known as endomysium. Clear? And many fibers, suppose this is a one fiber enclosed in an endomysium, then many fibers along with endomysium, they are wrapped in a other connective sheath is known as perimysium. Okay, one fiber is enclosed in a sheath known as endomysium, then many fibers again enclosed in another sheath is known as perimysium, and there are maybe number of perimysium 1, 2, and you can bring more, and then again covered with a next sheath is known as epimysium. Okay, so when you see the muscle, muscle fiber, all thinner fibers are encapsulated in, like you can see a packing box of pencil. If you take a big box, in big box there will be some smaller box, and you open the smaller box, there may be some other. So pencil, you can imagine pencil is a fiber, 10 pencil in one small box, then 10 small box in one bigger box, then 100 bigger box in one bigger cartoon. Clear? So same as here, muscle fiber, pencil. Clear? Many pencil in one box, small box is endomysium. Many endomysium covered in many endomysium along with some fibers covered in a big box is known as perimysium. And many big boxes are covered in a very big cartoon is known as epimysium. So as a gross, when you see muscle from outside like this, you will feel only and only endomysium. But when you cut, suppose if you see like this, okay, so you will see only endomysium. But when you see from here, if you see the electrical wire from this side, you will feel only one. But when you cut and see from the front, you will see there are number of fibers are originating. Same as with muscle. I will show you in diagram how they are structurally arranged, and this is the hierarchical arrangement. When you talk about the hierarchical arrangement of muscle is made up of protein, and basic building blocks are known as tropocollagen. Many tropocollagen unite, make one wrap. Is 1, 5, again 5, again more 5. And this type of arrangement is muscle. Okay. So individual muscle fiber wrapped and held together by multiple connective sheath, endomysium, perimysium, and epimysium. Epimysium is the outermost covering of the muscle, which contain maybe 1000 fiber, 10,000 fiber, whatever. Okay, depending on the girth of muscle. Suppose if you see here, this is the bigger muscle, it will have more amount of fibers in an epimysium, but if you see the finger muscle, it may have less amount of fiber in the epimysium. Clear? What is the function of all these components, all these structure? They are supporting. Suppose if you keep the pencil without box, pencil will go and here and there. Clear? But if you put all 10 pencils in a box, they will be structurally arranged and stabilized. So the main function of these three connective tissues are giving support and reinforcement. What is reinforcement? Supporting. I am backing you. So clear? Even I am standing here, but I am feeling relaxed. But if suppose if you imagine this is a 15th floor and there is no wall and I am standing here, I will not be feeling stabilized because I will afraid there is no support, no reinforcement. Clear? So if you stand on 25th floor, okay, and but there is a wall, you will feel secure. But there is no wall, your position is stable, but you may feel unsecure because there is no reinforcement. The reinforcement is a structure which support you from back. Clear? So these three connective tissues, epimysium, endomysium, and perimysium, they give a proper structure, a proper space in which the fibers are going parallelly. Clear? Now we will discuss one by one. Endomysium. What is endomysium? Surround each individual muscle fiber. Individual fiber, the one fiber. The one fiber is surrounded by your endomysium fiber, mainly reticular fiber. Reticular mean one fiber, then one fiber, one fiber. Like, this is like this. One fiber, again fiber. Clear? Next is epimysium. Outermost covering, I have told you, delts tissue surround entire muscle. Third is perimysium, fibrous connective tissue surround bundles, wrap muscle fiber. Perimysium, wrap bundles are fascicle. What is fascicle? Fascicle, suppose I have 10 fibers. All 10 fibers are surrounded by endomysium. Clear? But if you wrap 10 fiber with a thick covering, is known as perimysium, and whole structure covered by perimysium. Whole structure covered by perimysium is fascicle. Okay? One is known as fiber. One muscle cell. You know what is cell? Cell is the smallest unit of muscle. It may vary from very minute, even you cannot see, to a large, like nerve. Nerve is the largest cell. So if you see the muscle, suppose this is the whole cell. This is the whole cell, long cell, covered by a wrap is endomysium. And many muscle cell along with endomysium wrapped by other fiber is known as perimysium. Number of fibers surrounded by a tissue is known as, the tissue, perimysium is known as your fascicle. Okay? What is fascicle? A big box of pencil. 10 pencil, 10 boxes of 10 pencil. 10 boxes of 10 pencil arranged in one box is known as perimysium, or you can say it is fascicle. Fiber is one fiber. Fiber is one fiber. Fascicle is many fibers along with endomysium enclosed in a by perimysium is fascicle. Okay? First I told you fiber. The smallest, thinnest part of the muscle. Thinnest part of the muscle is muscle fiber. Okay? Many fibers, all fibers are attached, are wrapped by endomysium. And when you see 10 to 15, 20,000, 100 fibers, if they are enclosed in a layer known as perimysium, and whole structure is known as fascicle. Okay? Fiber, then fascicle. Now all connective sheath are continuous with each other and with tendon. Yeah, suppose if you are saying this is fiber, one fiber is enclosed by complete endomysium. Clear? and if you there is one other perimysium. So if you see the status of endomysium with perimysium, they are parallel and continuous. Endomysium is running over one finger, whereas perimysium is coming over the all fingers. Clear? So endomysium and perimysium, all connective tissue as well as they are parallel and continuous. If suppose this is the one fascicle, other fascicle, okay? When you wrap it with a complete is perimysium. So if you see the status of all, this is the endomysium, the parallel to endomysium is perimysium, the parallel to perimysium is epimysium. Okay? Epimysium is the outermost covering of the I can show you on my hand. Suppose this is if you cut the muscle like this. If you cut the muscle like this, clear? One big circle is epimysium. This is a perimysium, peri, and the structure which is covering this is smaller muscle fiber is endomysium. Clear? So when you see all are parallel to other and continuous. Clear? Muscle fiber contract. Muscle fiber, what are the muscle fiber? They are mainly actin and myosin. They have property of contraction, getting shortened and lengthened. All muscle fiber has a property of getting shortened and lengthened. When we have discussed about the proprioceptors, I have told you about extraphyseal fiber and intraphyseal fiber. Intraphyseal fiber are muscle spindle, whereas extraphyseal fiber are the main muscle fiber, the mainly actin myosin, which has capability of contraction. So muscle fiber is one made up of actin and myosin enclosed by endomysium. Contraction pulls on sheath. Suppose this is a fiber and one sheath is over this. Both are attached. This is a muscle fiber and this is a sheath. When it gets shortened, because it is attached to this, it will drag this. Clear? When two things are attached. When two things are attached, when inside portion, that is contractile filament, actin myosin, of which muscle fiber form. Muscle fiber forms from actin and myosin. Muscle fiber forms for actin and myosin, and when it contracts, because the upper portion of endomysium is attached with the muscle fiber, so it will contract. Clear? And when your endomysium will contract, because endomysium is again attached with the perimysium, your perimysium will contract. Clear? Suppose see here, if the inner one will contract, it will drag outer one. When this will contract, it will drag this one. When this contract, drag this one. Because all are attached like this, covering. When it shorten, it will drag this one, tendon. Clear? I will show you in diagram. Muscle fiber contract causes contraction pull on sheath. Contraction pull on sheath. What is sheath? Sheath is three: endomysium, perimysium and epimysium. So when inside a structure, they are mainly muscle fiber. Muscle fiber are surrounded by three layers. One layer, two layer, three layer. Okay. So when this will contract, if there is any damage in pencil, any change in pencil, it can affect your boxes. Same as when muscle fiber will contract, the outer covering will also pull. Muscle fiber, basically the thinnest unit of the muscle, complete muscle. Okay. And they have property of contraction, getting shortened. Suppose if you imagine here, muscle fiber, then endomysium, then perimysium, then epimysium, and your epimysium is going to attach, all are going to attach over the bone. When there will be contraction in one component, because all are parallel, fiber is going like this: above fiber endomysium, above endomysium perimysium, above perimysium epimysium. Because all are parallel. When there will be a shortening in one, it will pull. It will have a shortening in other also. Clear? So when because of shortening, they will drag the bone. So contraction pulls on sheath. Because of contraction in contractile element, that is muscle fiber, which is made up of actin and myosin, because of contraction in those contractile element, your sheaths, epi and endo and peri, they will also have some amount of pull on sheath. Clear? And sheath transmit force to bone, because your all sheath, your epimysium, perimysium and endomysium, they are going to attach over bone. Muscle fiber never attach over bone. Muscle fiber never attach over bone. Clear? Muscle fiber doesn't attach. They are inside the these three sheaths. The sheath is going to attach to bone, from bone to muscle. In between there is tendon. There is a tendon which doesn't have muscle fiber. Tendon is made up of all three: epimysium, endomysium and perimysium. Clear? They are elongated. They are elongated structure, and they are going to attach with the bone. But inside they are attached with the muscle. And when there is a pull, contraction in that fibers, they will pull the tendon, and tendon will pull the bone. Clear? And bones ultimately moves towards the direction of pull. If you suppose this is a fiber, this is a smaller one is a fiber, inside the fiber, and outer covering is little larger. When you pack anything, if you pack anything, the box should be little larger than the size of the pencil. Clear? So same as the outer covering, this endo, epi or peri, they are larger in size, lengthen than the fibers. So when they are going to attach, muscle fiber will not attach to bone, only your epi, endo and peri will going to attach to the bone. Okay? So when there will be any movement in the contractile element, it will have produced movement here, and because of movement over here, it will drag the bone. Clear? So this is the main function of your these three connective sheath. Muscle fiber will bring contraction, and because of contraction in muscle fiber, because they are attached with these sheath, they will pull some force on a sheath, and sheath will pull on bone, and bone will move. Clear? Now, what is the function of this three things? They are going to, there is two things: muscle origin and insertion. Ultimately, muscle is waste without bone, and bone is waste without muscle. That's why we always study musculoskeletal system. If we want to produce any movement, there should be healthy muscle as well as healthy bone. Clear? Without healthy muscle and healthy bone, one more component, nerve. All three work in conjunction, coordination. Without nerve, muscle cannot work. Without muscle, bone cannot work. So for proper and smooth function, we should have a healthy muscle, we should have a healthy nerve as well as a healthy bone. Clear? How muscle can produce, how muscle can move bone? Because muscle is going to attach ultimately over bone. Muscle is going to attach over bone, and muscle is attached over bone by two ends. Two ends. One is known as origin, another is known as insertion. Insertion is always towards the more mobile side. Suppose this is, if you see, this is your arm. Clear? See the biceps. Biceps is originate from scapula. Originate from scapula and inserted over the radial tuberosity of radius. So this, when you are moving this, see, in this movement, your scapula is not moving properly, but this bone is moving. So where the bone is moving is insertion. Okay? So insertion is always towards the attached side. Insertion is always attached towards the mobile side. So where bone is able to move more, muscle will be inserted over that area. Second is origin. Origin is always towards the less mobile side. Origin is always towards the less mobile side. The muscle has one origin, it has one insertion. Insertion should be always over the area where bone is able to move. It means near the joint. Where bone is able to move more, near the joint. And where bone is not able to move, somewhere in, suppose if you want to move the bone here, you cannot move because there is no joint. But if you want to move bone here, you can move. So insertion is always near the joint, whereas origin can be anywhere. So muscle has two components, one is origin and other is insertion. And by contracting muscle, by shortening the origin side toward the insertion side. Suppose if you see the biceps, your biceps is going to shorten toward the origin. This is the origin. Biceps originate from the coracoid process of scapula by two heads, long head and short head. And when you are going to flex your elbow flexion is most important function, because without elbow flexion, if suppose someone lost his elbow flexion, he cannot do any movement. If you want to eat, you cannot eat. Whatever movement, whatever flexibility you have at shoulder joint, if you want to comb, you cannot comb, because these all activities require your elbow flexion. Clear? So biceps originate from two heads, long head and short head, one from coracoid process and one the tip of scapula, and ultimately inserted over the. So you can see insertion is near the joint, whereas origin somewhere in the scapula. So always remember, origin is always in the area of more movement of the bone, and origin is always in the area of less movement toward the bone. And what is direct attachment? Epimysium is attached to bones periosteum. I have told you muscle fibers, they are basically contractile. They are not going to attach with bone. Muscle fiber are not going to. They are having some cover, and by means of cover they are going to attach with bone, because bone is harder structure. Bone is a harder structure and muscle fiber are very delicate structure. When you are going to attach some delicate thing with harder thing, it can damage. Harder thing can damage delicate thing. So muscle fibers, which are mainly actin and myosin, they are not going to directly attach with the bone. They have their epimysium, and extension of epimysium is going to attach with your bone. So this attachment is known as peri-epimysium. Peri-epimysium ki attachment. What is the outer covering of bone? Periosteal layer. Outer covering of bone is known as periosteum. So your epimysium or outer covering of muscle or tendon, you can say, is going to attach with the periosteum. It is the direct attachment. It is the direct attachment of muscle to bone. It means that muscle is directly attaching through tendon to the bone. Clear? This is known as direct attachment. But there is indirect attachment also. Suppose I want to capture you directly, hold you directly, it is direct. But if I am controlling him through you, it means indirect. So muscle is attaching to bone directly by means of epimysium, but muscle is attaching to bone indirectly by means of contact between your muscle fiber to endomysium to perimysium. Clear? Indirect attachment is smaller and more durable. Indirect attachment is smaller and more durable, because there is less chances of injury. There is less chances. Suppose if I hold you and I want to damage him, I cannot. But if I am directly holding you and want to damage, I can damage. So indirect attachment of muscle to bone is more durable, whereas direct attachment is less durable. Connective sheath extend beyond the muscle. Connective sheath extend beyond the muscle. As I have told you, suppose this is fiber, so your sheath will be beyond the sheath is covering. Fibers are actin myosin, so they are smaller. When they are going to match bone, they are not there. Only your outer covering is to attach the bone. Clear? That is why they are saying connective sheath extend beyond the muscle. They are rope-like structure. Rope-like means they are going to become a rope. If you hang anything over muscle, it can tear, because it is made up of fibers. It is made up of fiber. Fiber can tear easily, but anything like cord-like solid structure, it doesn't have flexibility, so it is more powerful. So your tendon is rope-like structure and sheet-like aponeurosis. Sheet means flat. So when your fiber are not there, the only membranes, epimysium, endomysium, and perimysium, so they will look like a flat sheet. Sheet means this is a rope-like structure. This is a structure. This is a rope-like. This structure, tendon, when tendon is attaching to bone in this shape, is rope-like tendon. But when your tendon is going to flatten and attach to the bone is flat sheath-like. Okay, this sheath-like tendon and this is the rope-like tendon. Circular tendon, it can be any type, either rope-like or sheath-like. Anchors muscle to connective tissue covering a bone, cartilage or fascia or another muscle. Clear? As I have told you, if your delicate structures are going to attach with harder structure, they can get damaged. But when muscles are using their coverings are harder because they are made up of connective tissues. Huh? So when their coverings are going to match with bone, fascia, cartilage, so their muscles are safer. Muscles are safer. Clear? When you are directly involved in any act, you can get damage. But when you are using someone to do the act, so you are safe. If there will be any injury, it will be to only him who is involved in the activities. Clear? Now come to microscopic anatomy of skeletal muscle fiber. I have told you the gross anatomy. When you are seeing the muscle from outside, many fibers wrapped in perimysium, then epimysium. It is outer side. But when you differentiate, when you analyze the one structure, one muscle, one fiber. Now I am going to see the microscopic anatomy of one fiber, long cylindrical cell. They are one muscle fiber. Now I am talking about only one muscle fiber. They are long cylindrical cell, 10 to 100 micron width. Their width is 10 to 100 micron. What is micron? You know? Micrometer. What is this? What is the value of micrometer? Micrometer mean 10 to power minus 6. When you divide 1 meter in 10 lakh, it means 1 million. When you divide 1 meter of an object into 10 million pieces, it is the width of one muscle fiber. The width of one muscle fiber, width mean breadth of one muscle fiber ranges from 10 micrometer to 100 micrometer. You cannot imagine how thin it is. How can? 1 meter is not more, and you are going to cut 1 meter weight into 10 lakh pieces. Clear? And the length of these fiber is very long, up to 30 centimeter. 30 centimeter is from here to here, around 1 feet. Okay? So length of the one muscle fiber will be very long. Length may be very long, but width is very narrow, very less. 10 micrometer. 10 micrometer mean very less, but length is long. So they are one cell. We all aware about cell. Cell is the basic structure of life. Cell is the basic unit of body. Huh? So, and we have different types of cell. Cylindrical cell, longer cell, columnar cell. So this muscle cell, also known as muscle fiber, also known as myofilament, mean muscle fiber. They have width of 10 to 100 micrometer and length of around 30 centimeter. Okay. Plasma membrane is termed sarcolemma. We have discussed about the different connective tissues which are encircling, which are giving protection to these fiber. But we all know all cell is made up, surrounded by a plasma membrane. That is the basic difference between plant and animal, because we are said to be alive, we are moving, we are having. What is the difference between plant and animal? Animal has property of movement, whereas plant doesn't have. And what is the basic difference between plant and animal? They have all the living things in their cell. When you see the cell anatomy of a plant, they have all structure which a animal cell have. But there is only one difference, which is the animal cell is enclosed by a living membrane. Whole structures of a cell are enclosed by a living membrane, whereas plant cell, all structures of plant cell are enclosed by a dead membrane. Clear? That's why plants are not able to move and we are able to move. except that outer covering, the other structure we had common. Whatever structure we have in our cell, they have. Whatever structure they have, we have. But the difference is the whole covering, outer covering. Plant is enclosed by a dead cell membrane known as cellulose, whereas an animal cell has a covering known as plasma membrane, which is alive. That's why we have all functional, whereas plant doesn't have. So I have told you muscle is a long cylindrical cell. Muscle is a long cylindrical cell with a width of 10 to 100 micrometer, as well as length may be up to 30 centimeter. So one cell, and the outer covering of that muscle fiber is made up of a membrane, plasma membrane known as sarcolemma. Like you can say cell membrane outer covering is plasma membrane, but the membrane which is covering the cell of muscle is known as sarcolemma. And the fluid which is filled inside that sarcolemma is known as sarcoplasm. Now, what is sarcoplasm? It stores large amount of glycogen. Sarcoplasm is a fluid which stores large amount of glycogen. Glycogen is a form of a glucose which is stored, which can be converted into glucose and give us energy. Large amount of myoglobin. So, I am discussing the content which sarcoplasm have. First is it has large amount of glycogen known as glycosomes. It also has large amount of myoglobin. Oxygen, what is myoglobin? Oxygen storing pigment similar to hemoglobin. Hemoglobin gene descend from duplicate ancestral myoglobin. Myoglobin is just like hemoglobin. Hemoglobin is a component of blood, which the main function of hemoglobin is carrying oxygen from the lungs to the tissues. Same as myoglobin is a structure present in cytoplasm and act as a source of energy. Now organelles. The usual organelles are present multiple nuclei beneath sarcolemma. Skeletal muscle are syncytia. Skeletal muscle are syncytia mean when skeletal muscle develop during embryonic stage of development. One cell develop from many cells, and that's why a single muscle cell, or you can say muscle fiber, has number of nucleus. Muscle cell is a multi-nucleate structure. It means when you talk about muscle cell, it will have mitochondria, it will have glycogen, it will have other important structure which a normal cell have. And whole fiber is known as myofibril, as well as sarcoplasm, sarcoplasmic reticulum, and T tubules. So these are the structure which a muscle fiber have. I am going to repeat that thing. First is glycosome, a source of energy. Myoglobin, a material which receive the oxygen, and the other things are they have multi-nucleus. They have myofibril as well as sarcoplasmic reticulum as well as T tubules. First we will discuss one by one. First is myofibril. Myofibril densely packed in muscle cell. They are the structure, they are the fibers which are densely packed in a muscle cell. More than 10 to power 5 per cell. It means around 1 lakh myofibrils are found within a muscle cell. Myofibrils, around 1 lakh myofibrils found within one muscle cell, and myofibrils form the 80% volume of the cell. I have told you cell has, what is a muscle fiber? It is a cell, muscle cell. It has myofibril, glycogen, it has T tubules, it has sarcoplasmic reticulum. So first we are discussing myofibrils. What is myofibrils? Many myofibrils densely packed in each muscle cell, more than 10 to power 5 per cell, and it makes the 80% of cell volume. It means that myofibril is the main component of muscle cell. It always run parallel to the length of cell. Myofibril always run parallel to muscle of cell. The diameter of myofibril is 1 to 2 micrometer. The diameter of myofibril is 1 to 2 micrometer and it contains contractile element. They are the fiber, main component of muscle fiber known as myofibril. The diameter is 1.1 to 2 micrometer and they form around 80% portion of your muscle cell, and these myofibril they are having contractile property. Now I am going to analyze what is the different structure, how we can make myofibril. When you see myofibril in a light microscope, you will find different bands are there. Different bands are there known as A band, also known as dark band. Light strip in center, S zone bisected by M line. All things are clear in the diagram. You can see in the diagram every zone. Myofibril, they are the main contractile unit, and they by their proper arrangement they are making different line, and these myofibrils are the basic functional unit of the muscle contraction. They have S zone which are bisected by M line, same as I band, they are light band, bisected by Z disc. Sarcomere, region of myofibril between two Z disc striation. What is sarcomere? One Z line this side, one Z line this side, dark band and light band. You can put two Z band at one side and other side, and in between there is light band formed by your I band, dark band formed by your M zone, M line, and both are all structure make one sarcomere. I am again going to repeat, sarcomere is formed by two Z line, in between there is light band formed by I band, dark band formed by M band, and complete form a one sarcomere. Region between myofibril and two Z disc is striation, repeating pattern of drive, and this pattern is repeated everywhere throughout the whole length of muscle. Suppose Z band, one Z band, two Z band, three Z band, four Z band. This type of repetition, one Z band, two Z band, three band, one Z band consists of I band and M band. I band is lighter band, M band is darker band. So throughout the whole length of muscle fiber, these arrangements are there. Near perfect alignment of band between myofibril give entire cell stripped appearance. Stripped means something in layers, something in layer. So when suppose this is I band, Z band, M band, I band, M band. So this type of appearance throughout the whole length give a stripping, layer wise arrangement of the filament. This can be explained by the diagram. You can see in the diagram. Now, what is the cause of myofibril striation? Banding pattern result from an orderly arrangement of thick myosin and thin actin filament. I have told you myofibril is consist of two things, actin filament and myosin filament. Actin filament forms lighter band, myosin form darker band, and they are arranged, and because of this arrangement they are forming a band. Thick filament extend entire length of A band. You can see in the diagram. Z disc composed of protein globulin. The Z disc which both Z disc form sarcomere. That Z disc is composed of a protein known as globulin, whereas your thin band is made up of a protein known as actin. Your darker band is made up of a protein known as myosin. What is the function of Z disc? What is the function of globulin in Z disc? It anchors thin filament, because your thin filaments are originating from your Z band, and Z band is made up of globulin protein. So by having a strong protein, it can anchor means support. It can support the thin filament. Now we are going to discuss second component, that is thick filament. Thick filament which forms the darker zone of the sarcomere. Sarcomere is the functional unit of your muscle fiber. How can we understand the contractile property of muscle? When we will be able to know the complete structure and mechanism of function of these I band, H band, M band and Z line. By seeing whole sarcomere function, we will be able to know how muscle can contract and how it can get relaxed. Now we are going to discuss about the anatomy of M band, which form the darker zone of sarcomere, composed of mainly approximately 200 myosin. That thick filament is made up of a protein known as myosin. Basically, they are rod-like structure, like a long rod. Two interwoven polypeptide chains. It means rod-like structure, but one rod interwoven with other rod like a rope structure. Like rope, you can see, you can take two cords and woven them, so they become like a rope-like structure. And basically, your myosin has a rod-like structure as well as central part of thick filament. It form the thick line, M line, you can say, or dark band. Next is globular head, present on end of thick filament. You can see in the diagram, in myosin there is one thick band, filament like structure, which is woven by two filaments, and you can see there are globules which are originating from different places of the myosin, and they are having present on ends of thick filament and cross bridges linking thick filament to thin filament during contraction. And these cross bridges are mainly responsible for making cross bridges with actin, and this is the basic of muscle. What is the basic structure which take part in muscle shortening and lengthening? So because of the cross bridge and the filament, both are having very crucial role in muscle contraction. Now we will discuss about the thin filament. Thin filament, that is actin, or you can say lighter zone. Lighter zone is made up of actin protein, possess site to which myosin heads bind during contraction. What happened? Myosin is a rod-like structure with globules in actin. It possesses sites to which myosin head bind during contraction. It means there is some site in that thin filament which can get attached from the myosin during muscle contraction mechanism. Also contain regulatory protein that is known as tropomyosin. Tropomyosin surround and stiffen actin core. What is the main function of tropomyosin? It surround and stiffen actin core. It means your tropomyosin is surrounding the actin core as well as block myosin binding site in relaxed muscle. Tropomyosin is the protein which are present over actin filament, and their main function is stiffening the actin core. And second main function, the main function in physiology of muscle is they can block the binding site. They can block the binding site in relaxed muscle. They are acting as a muscle relaxer. They can stop contraction. When they will activate it, muscle cannot produce contraction until unless they will not remove from there. Troponin, second protein of the actin. Actin has two proteins, tropomyosin and second is troponin. Troponin is made up of three polypeptide complex, T and I. It has inhibitory subunit. T and T help position tropomyosin on actin, and T and C binds to calcium. So we are discussing about actin protein, the second most important component of a muscle fiber. Actin protein forms the lighter zone of the sarcomere, and they are having two proteins. One is tropomyosin and other is troponin. The other component which are present in muscle fiber cell is sarcoplasmic reticulum. What is First we had discussed about myofibril. Myofibril is made up of two type of filament, actin and myosin. We have discussed in detail about the actin and myosin. The second structure which are surrounding that muscle fiber is sarcoplasmic reticulum. As you can see in the diagram, when it is elaborate, smooth endoplasmic reticulum. Interconnecting tubules loosely surround each myofibril. When you see in the figure, all sarcoplasmic reticulum are going to interconnect between two myofibril, regulate intracellular calcium level. This is the main function of sarcoplasmic reticulum. All your calcium which is required for muscle contraction. Generation of muscle contraction cannot take place without sufficient amount of calcium. That means there should be at least 10 to power minus 6 mole of calcium should be available at the end plate or the muscle fiber level to generate the contraction. But if there is no sufficient amount or this amount of calcium is present there, contraction cannot take place. We will discuss how these calcium play a role in muscle contraction in the muscle physiology. But you in anatomy should know all the calcium is stored in endoplasmic reticulum, which is just interconnecting tubules loosely surround each myofibril. Calcium releases on demand during stimulation, because without calcium muscle contraction cannot take place. During resting stage, calcium is stored within the sarcoplasmic reticulum, and when required, when you apply any sufficient amount of threshold to get muscle activated, it comes out from endoplasmic reticulum and sarcoplasmic reticulum and causes cross bridge formation between actin and myosin. So ultimately muscle will contract because of formation of actin and myosin cross bridges. Now next important structure of muscle cell is T-tubule. What is T-tubule? Invagination of plasma membrane, that is sarcolemma, elongated tubule penetrating cell anterior. Because we know there is a number of muscle fibers, and they all are contracted together. So how information of one fiber passes to other fiber? It is only because of presence of your T-tubule. T-tubule, the main function of T-tubule, it intimate contact with sarcoplasmic reticulum. Second, electrical impulses travel along sarcolemma, also along T-tubules. Whatever impulses comes from your motor nerve, they will travel to sarcolemma as well as through T-tubules to the other, from intercellular component or intramuscular area to outer area. Conduct impulses to deeper region of muscle. Suppose if some impulses come to your superficial tissues, it should go to the deeper level, and the deeper level of penetration of impulses is only because of presence of T-tubules in the muscle fiber. And the other function of T-tubule is transmit to every sarcomere. Whatever impulses is coming to one area can be transmitted to other areas of muscle fiber is because of presence of T-tubule. And impulse signal calcium release from SR, and by getting signal of impulses from T-tubule, the sarcoplasmic reticulum releases its calcium, and because of release of calcium it can cause muscle contraction. We will discuss in muscle physiology. So these are structures which are mainly responsible for muscle contraction. We had discussed about the muscle structure, their arrangement, their component, as well as we have discussed about their series elastic component as well as parallel elastic component. Parallel elastic component mean your epimysium, your perimysium and your endomysium. They are the outer covering of your contractile element of muscle. And what is your contractile element? They are basically actin and myosin. But I have told you actin and myosin, they never directly attach to bone, and when they are not directly attached to bone, they cannot produce any force over bone. So how muscle contraction can bring movement in bone? It is because of presence of these three connective tissues. First is endomysium, then epimysium, and then perimysium, and then epimysium. All fibers are enclosed in endomysium, and many fibers are enclosed in perimysium, and 1000 number of fibers are enclosed in epimysium, and all are going to attach with bone. So when there is any movement, mobility in your contractile element, that is actin and myosin, they will produce same effect over the connective tissues which are directly attached with the contractile element, and because of mobility in one, other will move, and because of movement of other tissues, they will move the bone toward the side of origin. Clear? So then the main contractile element is made up of a sarcomere. Sarcomere consists of two Z bands. In between there is light zone, dark zone. Light zone is made up of actin protein. Dark zone is made up of myosin protein. And when actin and myosin comes closer, they can cause contraction. When they go apart, they can cause relaxation. So by this coming and going of the closeness of actin and myosin, they can cause muscle contraction. So the major structural unit of muscle is sarcomere, and sarcomere, or you can say muscle cell. Muscle cell is mainly muscle fiber which it is surrounded by a plasma membrane like other cell. All cells are surrounded by all living cells are surrounded by living plasma membrane, but muscle cells are surrounded by sarcolemma, a membrane which confine whole cell. Sarcolemma has number of structure like mitochondria, like endoplasmic reticulum. You can say sarcoplasmic reticulum. Sarcolemma, same as there is some structure known as actin protein, myosin protein, nibulin, tannin, and all these proteins are responsible for muscle contraction. So we had finished the structure of muscle, and you can understand better by seeing the proper diagram which is given in the slide. Thank you.